So this next podcast deals with incomplete dominance and codominance. We would have learned about monohybrid crosses already. These are going to be monohybrid crosses that don't follow the strict laws of Mendelian inheritance. Just a quick note, the next three crosses that we do, the incomplete dominance, codominance, and then sex-linked traits, will never be dihybrid crosses for this course. So you only have to deal with them in monohybrids. So let's look first at incomplete dominance. With incomplete dominance, it's very simple. The F1 hybrids in incomplete dominance have an appearance that's in between the phenotypes of two parental varieties. So for Mendel, this would have been something that he never would have ever seen within pea plants. So if we look at snapdragons exhibit incomplete dominance. The homozygous condition is red, big R, big R. The homozygous recessive condition is white, little r, little r. If we cross these together, we find that all of them are heterozygous. And so the genotype is the same. So our genotype is the same as the other crosses. However, the phenotype is pink. So taking a red flower and a white flower makes a pink flower. And this is incomplete dominance because that trait is an in-between of red and white. So here's sort of the picture diagram showing this cross. If I take a red and a white, in the next generation, the F1 generation, I have pink, and I have 100% pink. They're heterozygous. When I cross two from the F1s, I find different ratios from what I would normally find from Mendelian traits that follow Mendel. Normally, Mendel would say that genotypically we have a 1:2 to 1 ratio, and phenotypically we would find a 3:1 ratio. That's Mendel. What we actually find with incomplete dominance is that genotype would still be a 1:2 to 1 ratio. However, the phenotype in this case is also 1:2 to 1. So in other words, 25% will be white, 50% will be pink, 25% will be red. That's incomplete dominance. Performing this right here, and remember, this was called a test cross. The test cross reveals to us that we actually find, by taking a pink and a white, we find 50% to be pink and 50% to be white. So anytime you see a heterozygous with incomplete dominance, you're going to end up with a different value. You'll end up with a different phenotype that will be somewhere in between. Usually a question will tell you that it's incomplete dominant, but just be aware that sometimes it doesn't, and sometimes you might have to figure that out. The easiest thing would be if you have 3 different colors, a white, a red, and then something intermediate, or a black, a white, and a gray. You probably got an incomplete dominant trait to work with in a problem. So that's incomplete dominance. Codominance is a little different. With codominance, we have 2 alleles that are expressed in heterozygous individuals. So in other words, you have 2 dominant alleles and you have 1 recessive allele. Normally, under normal Mendelian traits, you have a dominant and a recessive. And the dominant would be dominant, the recessive would be recessive. In codominance, we find that we have 2 dominant alleles. Now, the example that I'm going to use, and I'm not going to stray away from this in any of the problems that we do in grade 12 biology, is blood type, because blood type exhibits codominance. So the genotype for type A blood is, and notice I'm using the letter I, not A, because I'm dealing with an allele. I'm dealing with a dominant allele, and that dominant allele is A. The trait is I. So for type A blood, I can either be capital I, big A, capital I, big A, or I can be capital I, big A, little I. This is the recessive allele, and this is the dominant A allele. Now, if you're type B blood, you're pretty much going to be the same, except they'll be Bs. If you're type AB blood, then your genotype looks like this. So you have the capital A, I allele, but you also exhibit the B allele. And if you're O, you are the homozygous recessive. Okay, now on a red blood cell, very quickly, what this looks like is this. On a red blood cell, we find antigens. And those antigens have particular shapes. Now I'm going to make it as cartoony as possible just so that you understand. Type A blood, the shapes may look like triangles on a red blood cell. And so if we had type A blood, we would have blood cells with little triangles on it. Type B blood may have little squares. And so if we had any form of type B blood, we would have little squares. Type AB blood would have both. So not only would we have the triangles, but we would also have the squares. And what these really are, what these antigens do is they allow the red blood cells to recognize themselves. If they don't see themselves, it's an invader. And then there's a defense mechanism, which is blood clotting. If you're little I, if you're homozygous recessive, then your red blood cells have no antigens on them. They have no squares or no triangles. So that's the basics to codominance. When we deal with codominant problems, we have to set it up using the I's as our primary and using superscripts for our actual trait. So if we have a homozygous male type B, his genotype, because he's homozygous, will be IB, IB. If we have a heterozygous female that's type A, she will be IA, little I. And if we were to perform this cross to see what sort of children they would get, right? Remember, we're only going to be using Punnett squares, that are monohybrids. We don't use dihybrid Punnett squares. It would be very complicated. I mean, we could, but we're not going to. So what I would find genotypically is that 50% of the offspring will be type AB, and 50% will be IB, little I. Phenotypically, what this means is that 50% are type AB, and 50% will be type B. Remember, phenotype is what we see, and so that's what we would see in a blood type. If we had a male type O and a female type AB, I would like you to pause and figure it out, and then I'll reveal the solution. Okay, you should have a solution. So let's have a look. Our parents, remember, always start with the parents. So one parent is type O, and we have a female that's type AB. Setting up the Punnett square. It would look something like this. Filling in the Punnett square, you should have had this. Right? Doing our genotypes, we would find 50% would be A little I, 50% IB little I. Phenotypes, 50% type A, and 50% type B. So that's what we should have gotten with this particular problem. Our last problem, again, if a boy has blood type O and his sister has blood type AB, what are the genotypes and phenotypes of the parents? Again, pause. try to come up with an answer, and then hit play and we will do the solution. Okay, hopefully you figured this one out. In this particular problem, we actually have to go backwards. I know the children in this case, so there's a boy that's got type A or type O, and we got a sister that's got type AB. Again, because it's not sex-linked, I don't have to worry about accounting for boys or girls. So what I would do with this is set up a Punnett square and put these children in. So I know that one of the boys, the boy itself, is that. I know that one of those little i's came from mom and one of those little i's came from dad. The girl was that, type AB, so she would actually be IAIB, sorry. So I know that one of those letters, if it was A, doesn't matter really, came from dad, and one of those letters came from mom. And remember, the question doesn't give us specifically what the parents were, but I know that by going backwards that I have one parent that is this, and I have one parent that is that. You should have enough information now to go on to do the incomplete dominance and codominance problems, and I'm going to have a different podcast for sex-linked traits.